Hi, welcome to the last topic for the first grading, and that is the enzymes. For this topic, you will be able to describe the components of an enzyme, explain the oxidation reduction reactions or redox reactions, and determine how factors such as pH, temperature, and substrate affect enzyme activity. So before we proceed with the attainment of our objectives, let us view videos that will serve as a recap of the topics that you had discussed in your junior high school. And let us start with proteins.
So far, we've covered two of the organic macromolecules, carbohydrates and lipids. Today, we'll be talking about proteins. Proteins have many uses. For example, many different proteins are used to form the majority of various body tissues, including muscle, bone, cartilage, and hair. Other proteins form specialized channels in cell membranes to let certain substances into or out of the cell. Immunoglobulins, also known as antibodies, are made of proteins. They are an important part of the immune system. Antibody proteins label foreign substances in the body so that the immune system can identify and destroy them. And all living organisms use regulatory proteins called enzymes to speed up chemical reactions in the body. So, what are proteins made of? Well, all proteins are organic macromolecules, which means they are large molecules containing the element carbon. Like carbohydrates and lipids, proteins also contain hydrogen and oxygen. In addition, proteins also contain the element nitrogen. So what is the structure of proteins? Well, the protein building block, or monomer, is something called an amino acid molecule. Every amino acid has a particular structure. They all have the same amino group on one side and a weak acid called a carboxyl group on the other. But each amino acid also has a unique side chain called an R group. It's made up of one or more atoms that distinguish one amino acid from another. There are only 20 possible different R groups, so that means there are only 20 possible different amino acids. A protein is created when amino acids chemically bond to each other. The bonds that connect amino acids to make a protein are called peptide bonds. Small protein molecules are sometimes referred to as protein polymers or peptides. Like interconnecting toy blocks, amino acids can join together to form even longer protein polymers called polypeptides. Changing the order of the amino acid sequence, or the length of the polypeptide, is what creates countless different proteins. In addition, a protein's final folded shape is critical to the function it performs. Heat or chemicals can change a protein's shape, but not its amino acid order. However, the change in a protein's shape can cause the protein to lose its functionality. This is called denaturing a protein. Denaturing is usually not reversible. An example of denaturing a protein happens when you fry an egg. Egg whites are pure protein. If you heat an egg, the protein in the egg white becomes denatured. You can see the result of this denaturing as the egg white changes from a clear-colored liquid to a white-colored solid. Even after cooling, the egg white's denatured proteins can't change back to their original shape. So, the egg white retains its opaque white color and solid form. To sum up, proteins are organic macromolecules containing carbon, hydrogen, oxygen, and nitrogen. Enzymes, most body tissues, and many cells in the immune system are all made up of proteins. Amino acids are the monomers that make up proteins. There are only 20 different amino acids. Small protein polymer molecules are often called peptides. Long chains of amino acids are also a type of protein polymer called polypeptides. Proteins differ in amino acid sequence, polypeptide length, and folded shape. A protein's folded shape is critical to its function.
After proteins, we are heading next to nucleic acids.
So far, we've covered two of the organic macromolecules. The final organic macromolecule we'll cover is nucleic acids. A nucleic acid is an organic macromolecule, which means it's a large organic compound made from thousands or hundreds of thousands of smaller molecules. There are two types of nucleic acids: deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. Notice that both terms actually contain the words nucleic acid in them. In living organisms, DNA is the main component of chromatin, which will condense into the familiar chromosome shape prior to cell division. The DNA in chromosomes is organized in a specific order that makes up an organism's genes. Genes contain the directions for every function, trait, and activity in a living organism. These activities include growth, reproduction, and especially heredity, which means the ability to pass on genes to offspring. The other type of nucleic acid, RNA, is integrally involved in building specific proteins by assembling their amino acids in the correct order. Proteins are responsible for all cellular functions in living organisms. So, what are nucleic acids made of? Of course, as an organic macromolecule, nucleic acids contain the element carbon. Like proteins, nucleic acids also contain hydrogen, oxygen, and nitrogen. But nucleic acids also contain the element phosphorus. So, what is the structure of nucleic acids? Well, both DNA and RNA are made up of monomers called nucleotides. All nucleotides contain 3 compounds. The first compound is a phosphate group, which contains phosphorus. The second compound is a 5-carbon sugar. All 5-carbon sugars can be called a pentose. A good way to remember this is that pent means 5, and the suffix -ose means sugar. The specific pentose sugar in RNA is called ribose. The pentose sugar in DNA has one less oxygen than ribose in RNA, which leads to its name deoxyribose. The third compound in a nucleic acid nucleotide is a weak base that always contains nitrogen. For this reason, this compound is referred to as a nitrogenous base. The nucleotides in DNA contain one of four possible nitrogenous bases. They are called adenine, thymine, cytosine, or guanine. These bases are usually abbreviated as A, T, C, and G. Similarly, the nucleotides in RNA also contain one of four nitrogenous bases, adenine, uracil, cytosine, or guanine. They are abbreviated as A, U, C, and G. Notice that both DNA and RNA have adenine, cytosine, and guanine, but only DNA has thymine and only RNA has uracil. So, how do nucleotide monomers assemble into nucleic acids? Well, the phosphate group in one nucleotide bonds with the pentose sugar in another nucleotide. The assembled string of nucleotides forms a nucleic acid polymer. Even though both RNA and DNA have alternating sugar and phosphate groups, there are differences in their overall structure. For example, RNA is a single-stranded nucleic acid, while DNA is a double-stranded nucleic acid. The two strands in DNA are linked by a hydrogen bond that connects the nitrogenous bases from one strand to the nitrogenous bases from the other strand. In summary, nucleic acids are organic macromolecules. The two types of nucleic acids are DNA and RNA. DNA and RNA are used to transmit hereditary information and to instruct the cell how to properly construct proteins. Nucleic acids contain carbon, hydrogen, oxygen, nitrogen, and phosphorus. Nucleotides are the monomers that bond together to form DNA or RNA. Nucleotides consist of three compounds: a phosphate group, a 5-carbon sugar, also known as a pentose, and a nitrogenous base. The nitrogenous bases in DNA are adenine, thymine, cytosine, and guanine. The nitrogenous bases in RNA are adenine, uracil, cytosine, and guanine. The 5-carbon sugar in DNA is called deoxyribose, while the 5-carbon sugar in RNA is called ribose.
Okay, I hope our minds are refreshed by the review of some of the organic macromolecules. So today we will discuss the following: what is an enzyme, what is a redox reaction, and how factors or certain factors affect enzyme. So our first lesson is the components of an enzyme. The previous video is very much important because, as you can see, enzymes are proteins or can be RNA. So that play a critical role in controlling and speeding up chemical reactions in the human body and other living organisms. They do this by lowering the activation energy required for a certain reaction to occur, making it happen more quickly and efficiently. Enzymes have a specific binding site called the active site, which is uniquely shaped to interact with specific substances. Enzymes can have cofactor such as metal ions or coenzymes like vitamins, which help them perform their catalytic functions. So when an enzyme is bound to its coenzyme, it forms a holoenzyme. It is an enzyme without its enzyme. It's referred as a apoenzyme. So if an enzyme is bound with a coenzyme, it is called holoenzyme. And if the enzyme without its coenzyme is apoenzyme. So there are two main models to explain enzyme action: the lock and key hypothesis and the induced fit. So what is the difference between the two? Lock and key. So lock and key. So this model suggests that the substrate fits perfectly into the enzyme's active site. So it is just like how a key fits into a lock. While the induced fit hypothesis, the enzyme changes its shape upon binding to the substrate, ensuring a tight fit and enhancing the reaction. So enzyme activity can be influenced by various environmental factors including temperature, pH, enzyme concentration, and substrate concentration. Enzymes work optimally at specific temperatures and pH ranges, and their activity can be inhibited by factors such as extreme temperature or pH levels. Inhibitors are substances that can block or inhibit or reduce enzyme activity. So there are two main types of inhibitors: the competitive inhibitors and the non-competitive inhibitors. So the difference between the two is that the competitive inhibitors occupy the enzyme's active site, preventing the substrate from binding, while the non-competitive inhibitor are attached to other parts of the enzymes, causing a change in its shape and inhibiting its activity. So to further discuss the enzyme components, I prepared a video for you to watch. Uy, the audio is not turned on. Hold on, class. Please excuse for the technical delay.
Enzymes. Human body is a product of different chemical reactions and processes. But what controls these reactions? In 1833, a French chemist, Anselme Pien, was the first to discover the vital force that drove these reactions and named it enzyme. Enzymes are substances, proteins, or in some cases, ribonucleic acid, RNA, that speed up a biochemical reaction by modifying specific substances called substrates. Enzymes are supremely selective in who they bind to and modify, hence their specificity. Mode of action. Easier said, but how does a tiny enzyme speed up a chemical reaction? The answer to this is simple. Enzymes perform this critical task by lowering a reaction's activation energy, that is, the amount of energy needed for the reaction to begin. Enzymes bind to their substrates, hold them in such that chemical bond breaking and bond forming processes take place more easily. Enzymes have a specific place in them called active site, where the substrate binds and real-time action takes place. Active site has a specific size, shape, and chemical behavior rendered to it by specific arrangement of amino acids. Thanks to these amino acids, an enzyme's active site is unique only to a particular substrate. In addition to the active site, many enzymes consist of a non-protein part called cofactor. Cofactors may be cations, positively charged metal ions, activators, bound temporarily to the active site to activate the enzyme, organic molecules, Vitamins or vitamin products, coenzymes, that join enzyme-substrate complex temporarily. Prosthetic groups, permanently enzyme-bound. Many enzymes only perform their catalytic role when associated a coenzyme. An inactivated enzyme, protein, along with its coenzyme, non-protein, makes up a system called holoenzyme. To add to the complexity, scientists termed this inactivated enzyme as apoenzyme. And therefore, the equation becomes holoenzyme equals apoenzyme plus coenzyme. Models of enzyme action. Lock and key hypothesis. This is the simplest model to represent how an enzyme works. The substrate simply fits into the active site to form a reaction intermediate. Just like the key fits in its specific lock, the shape isn't changed here. Rather, the structure of the substrate absolutely complements the structure of the enzyme, like puzzle pieces. Induced fit hypothesis. In this model, the enzyme, upon binding of substrate, changes shape. The matching between an enzyme's active site and the substrate isn't just like two puzzle pieces fitting together. Rather, the enzyme changes shape and binds to its substrate even more tightly. This fine-tuning of the enzyme to fit the substrate is called induced fit. Environmental effects on enzyme function. Active sites are very sensitive. They sense even the slightest change in the environment and respond accordingly. Some of the factors that affect the active site and consequently enzyme function include temperature. The suitable temperature for enzymes to function properly is 37 degrees Celsius. Increasing or decreasing the temperature above 37 degrees Celsius affects chemical bonds in the active site, making them less suited to bind substrates. Higher temperatures denature enzymes. pH. Amino acids present in the active site are acidic or basic. Fluctuation in pH can affect these amino acids, making it hard for substrates to bind. Extreme pH values can denature enzymes. Enzyme concentration. Increasing enzyme concentration will increase the rate of reaction, as more enzymes will be available to bind with the substrates. However, after a certain concentration, any increase will have no effect on the rate of reaction. Substrate concentration. Increasing substrate concentration increases the rate of reaction. This is because more substrate molecules will be colliding with enzyme molecules, so more product will be formed. But again, this effect is valid up to a certain concentration. Inhibition of enzyme activity. Some evil substances called inhibitors reduce or even stop the activity of enzymes in biochemical reactions. They either block or distort the active site, thus inhibiting the reaction. Based on this, there are two types of inhibitors given below. Competitive inhibitors occupy the active site and prevent a substrate molecule from binding to the enzyme. Non-competitive inhibitors attach to parts of the enzyme other than the active site to distort the shape of an enzyme.
Okay, to distort the shape of an enzyme, so that is a non-competitive inhibitor. After viewing the video, I know that you are now excited to answer the following questions. So what are enzymes? How does a tiny enzyme speed up a chemical reaction? What is an active site? What are the models of enzyme action? And what are the factors that affect the active site and enzyme function? So let us now check your answers for number 1. What is an enzyme? Anyone? Okay, an enzyme are substances, proteins, or in some cases RNA that speed up a biochemical reaction by modifying specific substances called substrates. Number 2, how does an enzyme speed up a chemical reaction? It is what? How? Enzyme perform this critical task by lowering the reaction's activation energy, and that is the amount of energy needed for the reaction to begin. How can the enzyme do this? By binding to their substrate. Hold them in such that chemical bond breaking and bond forming processes can take place more easily. 3. What is an active site? Is where the substrate binds and real-time action takes place. Active site has a specific size and shape and chemical behavior rendered to it by specific arrangement of amino acids. Thanks to these amino acids, an enzyme active site is unique only to a particular substrate. 4. What are the models of enzyme actions? What are those models? We have 2. The lock and key hypothesis, the simplest model to represent how an enzyme works. Substrate simply fits into the active site to form a reaction immediate, just like the key fits in its specific lock. The shape isn't changed here, rather the structure of substrate absolutely complements the structure of the enzyme like a puzzle. So fit like a lock and key. And the second one is the induced fit, where the enzyme, upon binding of substrate, changes shape. The matching between an enzyme's active site and the substrate isn't just like two puzzle pieces fitting together. Rather, the enzyme changes. Si enzyme na ang nag-adjust sa shape and binds to its substrate even more tightly. This fine-tuning of the enzyme to fit the substance is called induced fit. The fifth question, what are some of the factors that affect the active site and enzyme function? What are those factors? 1, we have the temperature. Suitable temperature for enzymes to function is 37 degrees Celsius. Increasing or decreasing the temperature above 37 affect chemical bonds in the active site, making them less suited to bind substrate. Higher temperature, denature the enzyme. So if you have fever, it is good that you have a fever. Why? A fever is like a signal to your body that something is wrong. However, if the temperature is really high, it will be harmful to you because the higher the temperature, denature the enzyme, so your processes will halt or there will be problem on your body mechanisms because your enzymes are denatured. Next is the pH. Amino acids present an active site are acidic or basic. Fluctuation in pH can affect this amino acid, making it hard for substrates to bind. Extreme pH values can denature also the enzyme. Very high in acidity or very basic can denature the enzyme. Next, the enzyme concentration. Increasing enzyme concentration will increase the rate of reaction as more enzymes will be available to bind with substrate. However, after a certain concentration, any increase will have no effect on the rate of reaction. Okay, substrate concentration. Increasing substrate concentration increases the rate. This is because more substrate molecules will be colliding with enzyme molecules, so more product will be formed. And the last question, what inhibits of enzyme activity? What are those inhibitors? We have the competitive inhibitors and non-competitive inhibitors. So, competitive, occupy the active site and prevent the substrate molecule from binding to it. O nangilog, wala siya. Kanang, iya dyan secure ako, ra dyan ni, di dyan ni pwede mailog sa lain. Okay? While the non-competitive inhibitors, they attach to the parts of the enzyme other than the active site to distort the shape of an enzyme. Next lesson is the redox reaction. So, redox reaction, redox. In chemical reactions, the movement of electrons from one atom to another is very important. It is crucial. So, oxidation, what is the meaning of oxidation? Refers to the process where an atom loses, oxidize, while reduction involves gaining. So, it is very opposite. Oxidation loses, reduction gains electrons. Okay, so to remember this, we have mnemonics oil rig. Oxidation is loss oil. Rig reduction is gain. Okay? So in the context of redox reaction, the atom that loses electrons acts as reducing agent, while the one gaining electrons acts as oxidizing agent. Okay? So this redox process ensures the overall oxidation numbers balance out to zero, highlighting the transfer of electrons between reactants in the chemical reaction. So let us now watch a video about redox reaction.
In this video, we will be talking about redox reactions or the oxidation-reduction reactions and its practical application to the real life. Redox reaction involves the movement of electrons from one atom to the other. It means one atom is actually losing electrons while the other is gaining that electron. Oxidation means that the atom is losing electrons, while reduction means that the atom is gaining electrons. To easily remember this pattern, just be familiar with the mnemonics oil rig. When an atom loses electrons, it is said to be a reducing agent, while when it gains electrons, it is said to be an oxidizing agent. So this might be confusing again, so remember the mnemonics Leora and Giroa. Losing electrons is oxidation, so it is a reducing agent, while gaining electrons is reduction, so it is an oxidizing agent. In this diagram, atom A loses electrons, while atom B is gaining that electron. So atom A is oxidized, while atom B is reduced. Therefore, atom A is a reducing agent, while atom B is an oxidizing agent. One practical application of this is the photosynthesis process happening in plants. Photosynthesis happens when carbon dioxide and water, together with the light energy, interacts to form carbohydrate or the glucose plus oxygen. It turns out that in this chemical equation, there are two reactions of photosynthesis taking place, the light reaction and a dark reaction. For the first step, we have a light reaction taking place when the oxygen in water is oxidized by the light energy. H here represents a reducing agent. For the step 2, we have the hydrogen reduces the carbon in carbon dioxide, a type of dark reaction. Since oxygen has a -2 oxidation number, and the oxidation numbers of a certain molecule should always be equal to 0, so it means that the oxygen has a total of -4, leaving carbon with a +4 oxidation state. We can see that its oxidation state turns from +4 to 0. Hydrogen has an oxidation state of +1, making the hydrogen here +2 and the oxygen -2, leaving everything else 0. So in this application, we can see that photosynthesis is undergoing a redox reaction.
Okay. So now we will proceed to our last topic, and that is the factors that affect enzyme activity. So what are these factors? In chemical reactions, energy changes occur and reactions can be either endothermic. Endo means to absorb energy, or exo, to release energy. Exothermic, release energy. Endothermic, absorb energy. So the peak energy point in a reaction is called the activation energy, the minimum energy required for a reaction to take place. So to speed up reactions, catalysts can be used. So catalysts are substances that lower the activation energy without being consumed in the reaction. So they make reactions more efficient and faster. Catalysts are particularly important. Yes, it is really important in biological reactions when there are known as enzymes. So we can really associate the word enzyme with the word catalyst. Speeds up chemical reaction. Enzymes are protein-based biological catalysts, each with a unique active site and fits a specific substrate, forming an enzyme substrate complex. So enzymes lower the activation energy, making reactions occur more quickly. And as part of the discussions that we had, enzymes are crucial in living organisms. They work optimally at specific pH levels and temperature. Higher and lower than its normal pH level and temperature will denature the enzymes. So we have factors like pH, temperature, and the presence of other chemicals can impact in enzyme effectiveness. Enzymes are essential for many biological processes and allow reaction to happen at the right rate within the body. So, to understand more about these factors, let us view this video.
Today, we're going to talk about catalysts and enzymes. To understand enzymes, we need to know that chemical reactions require energy to occur. Recall that chemical reactions break the chemical bonds in reactants and rearrange those bonds to make products. Let's look at an energy diagram to see how energy changes as a chemical reaction progresses. At the start of a reaction, the reactants will have some amount of energy. If the products end up with more energy than the reactants had at the beginning, then this means the reaction absorbed energy from the environment. This is called an endothermic reaction. However, if the products end up with less energy than the reactants, then this means the reaction released energy. This is called an exothermic reaction. Either way, notice the peak in the energy hill just before the products begin to form. This peak represents the minimum energy the reactants required for the reaction to take place. The reactants need this energy to break their chemical bonds so that different bonds can be formed to make new products. This minimum amount of energy required for a reaction to take place is called activation energy. If the activation energy isn't reached, no reaction takes place. Sometimes, scientists need to make a reaction happen faster. If you want to speed up a reaction, you could increase the energy of the reactants by increasing their temperature. Or you could add something called a catalyst. A catalyst is a substance that catalyzes or speeds up a chemical reaction without it being changed or used up by the reaction. Catalysts work by lowering the activation energy needed for the reaction to take place. By requiring less energy, catalysts make reactions happen faster and more efficiently because more of the reacting particles are likely to have sufficient energy or activation energy. And the higher the catalyst concentration, the faster the reaction takes place. Since catalysts aren't changed or used up by a reaction, they're available to catalyze more reactants. Chemical reactions are done in labs. But many chemical reactions also happen constantly inside your body. In living organisms, many reactions require catalysts. Biological catalysts are called enzymes. An enzyme is always made up of proteins. Proteins are one of the 4 types of organic compounds. We'll discuss them in more detail separately. Enzymes make life possible by lowering the amount of activation energy needed and therefore speed up reactions that would otherwise take too long. You need enzymes to stay alive. A reactant acted upon by an enzyme is called a substrate. Each enzyme has a unique active site that only a certain substrate can attach to, like a very specific puzzle piece. When attached, they form what is called an enzyme-substrate complex. This fit between an enzyme and substrate is so specific that it's often compared to a lock and key. When the substrate attaches to the enzyme lowers the activation energy needed to break the bonds in the substrate. Once the bonds break in the substrate, it separates into multiple components called products, which leave the active site. Afterward, the enzyme is still available to participate in another reaction with the same type of substrate. So, what factors affect how well an enzyme works? Well, enzymes work best around particular pH levels and certain temperatures. And the presence or absence of other chemicals can also impact their effectiveness. Not surprisingly, humans and other organisms have adapted to use enzymes that are most effective at the organism's natural temperature and pH values. To sum up, chemical reactions require energy to break the reactant's chemical bonds. The minimum amount of energy required for a reaction to happen is called the activation energy. Catalysts speed up reactions by lowering the activation energy. Catalysts aren't used up by chemical reactions. Biological catalysts are called enzymes. Enzymes are made of proteins. Enzymes act on reactants called substrates. Substrates bind to unique locations called active sites on enzymes. And factors that affect how well an enzyme works include pH, temperature, and other chemicals.
And that's the end of our topic on enzymes. So we are already at the end of our first quarter lessons. So your task will be to analyze or to finalize your notes and study for your examination. God bless, and I wish for your success in your exams.